The webinar room. How are you all doing today? Welcome in. We're going to get started in just a minute or two. I just want to make sure that we get started with everybody in the room that's going to be joining us today. But while we have a moment, if you notice on the right-hand side, there is a chat box. If you can just go ahead and I'm going to switch one thing here. Just say hello to us. Tell us what your name is and where you are logging in from. We would love to say hello to you today. Hi, Jennifer. Hi, Marianne. Donna. Hey, Vince. It's good to have you all here today. We are in the Pacific Northwest in Washington. It's a rather rainy day today, but it is beautiful, as Momo and I were discussing. But it was sunny yesterday, so woohoo, awesome. Hey, Larry. Hi, Leslie, Grant, Xiaoling, Camellia, Abby. Oh my gosh, it's going so fast, Momo. I can't say everybody's name. It's pretty incredible. Welcome everybody into the room. Cynthia, Yashas, William, Kathy, Sybil, Dr. Mark, Jeff, Emma, Kristen, Michael, and Michael's from UK. Let's see where else we got. Amsterdam.
Argentina.
Yeah, nice. Very good. And we have somebody from Bellingham who's just like our neighbor, just like an hour or two up north. Yeah, Ricky, represent Washington. Excellent, excellent.
Wow, UAE.
Where? Ooh.
Oh, UAE and Ireland and Cyprus.
You're going to get back in the UAE pretty soon, aren't you?
I'm going next week, yes.
Ooh, nice. Very nice. Excellent. And Costas from Greece. Nice. And then Canada, of course. So we're representing all around the world today, which is totally, totally awesome. And Canada again. Lisa, Los Angeles. No, hey, Los Angeles is wonderful. I lived there for a very long time. And Larry, San Jose, Martha, Arizona. I think we can get started. There's probably going to be people still filtering in, but that is totally okay. Welcome to all of you today. This is our monthly live Viome topical content webinar where we love to talk all things nutrition, health, wellness, and of course, Viome. And today our focus is on what makes Viome different and unique, as well as how the science and technology teams at Viome are making discoveries and advancements that are influencing the future of healthcare as we know it. And I am Lisa Shomo. I'm head of community at Viome. I run our live events and community programs. And today I have an extra special guest with me. He is an entrepreneur scientist and a trailblazer in microbiology and biochemistry. He is our founding chief science officer and head of clinical research, Dr. Momo Vuyisich. Welcome.
Thank you, Lisa. Thank you. Excited to be here.
Yes. Now, I have asked Momo to come onto our broadcast this month because I thought it was time. It was well past time to talk about the Viome difference, and he agreed. Thank goodness. And I want to jump right in, Momo. Viome is such a unique entity, and I'd love for you to talk about that just a little. How are we able to set ourselves apart from all other microbiome health and wellness brands?
Happy to discuss. This is a very important topic, and we'll also, very importantly, later on dive really deeply into microbial physiology and how it interacts with human physiology and how your immune system is actually to some degree regulated by the bacteria and so on. So I'm really excited about this presentation. All right, well, so there are foundational differences between Viome and everyone else, and then there are technological differences between Viome and everyone else. So let's talk about foundational differences. As an academic in my previous career, I realized that neither academia nor pharma/healthcare system are actually solving the problem that I want solved, which is I want to identify the root causes of chronic diseases and then literally prevent them and cure them all so that we can treat chronic diseases and cancers like we treat the Black Plague from the 13th century. We don't have Black Plague anymore. We know what causes it. We know how it's transmitted. We know how to prevent it, how to cure it, and it's no longer a concern. Why can't we apply the same thing to multiple sclerosis and Alzheimer's and Parkinson's and all types of cancers? We can. We have all the technology, so let's do it. But unfortunately, there are no entities out there that are systematically doing it. So that's why we created Viome, to serve humanity and to solve the problem of root causes of chronic diseases and cancers. So that's the foundational, difference. The technological difference is what we're going to discuss through this presentation. So I think most of you know this, so I'm going to skim through this, but basically we are getting sicker as a nation, and the numbers are absolutely not acceptable. So there's $4.1 trillion spent per year in the United States. I'm sure there's a lot of healthcare expenditures in other countries, but just not as much as in the US. And 90% of these expenditures are spent on chronic diseases and management of chronic diseases, because we can't cure chronic diseases. There is no known cause. And in the US, the epidemic is hitting 60% of US adults. And that's just unacceptable, because we literally today have all the science and technology to learn what are the root causes and prevent and cure chronic diseases. We just need to focus on actually doing it.
Right.
One more example I want to give here is that type 2 diabetes is one of the most common chronic diseases in the world, and yet it's increasing in its prevalence. And so what you can see, or incidence, so what you can see here in gray graphs is the current status of type 2 diabetes for different age groups. And what you can see in the orange graph is the incidence, meaning new cases. And new cases are actually growing in younger populations and in countries that traditionally have not seen a high disease burden. So this is a growing problem, and it turns out that diabetes is one of the more costly and more painful sort of a chronic disease as it progresses throughout the years.
Momo, actually have a question. So a friend of mine who is younger, as you were just saying, just got diagnosed with type 2 diabetes. And so her doctor told her that it's genetic, and there's really not much else that she can do for it except take the medication to manage it. So what do you say to that?
Well, there's a small genetic component to type 2 diabetes and many other chronic diseases, but it's a small genetic component. But certainly that's not a death sentence, and that's not your fate. You can clearly, it's possible to change the course of many chronic diseases with diet and lifestyle changes. We just need to learn exactly how to do that. But for diabetes, there are even some general guidelines for diabetes and obesity and metabolic diseases. There are some general guidelines you can follow without any fancy tests, and you can still heavily impact the progression of these diseases, and you can even reverse them. So genetics is just a small part of it.
Good to hear.
All right, so let's go into genetics. Look at that. The next slide is about, so, 10 years ago, let's say, when I told people that genetics plays a minor role in chronic diseases, they said, well, there's no evidence for that, and genetics is important, and we should study genetics. Well, today, luckily, it has been revealed in very, very nicely carried out genetic twin studies that genetics plays a minor role in essentially all chronic diseases. And so there are some really nice publications that I listed here that talk about how genetics plays a minor role in chronic diseases and cancers. And it's the environment, meaning your microbiome, your nutrition, toxins you're exposed to, all of those things that are actually much more important in the onset and progression of chronic diseases. So I would encourage the audience to read about this. This is a huge topic we simply cannot cover, but for now, just a summary that genetics plays a minor role, and that you can actually change your fate with your diet and lifestyle very heavily.
So I actually had another thought. So we have two very large systems here, academia and pharma. And then they're supposed to be solving these problems, right? So what do you have to say on that?
Well, they're actually not supposed to be solving this problem. We think from reading the news and from basically just being told that they're working on these problems, but they're in fact not focused on these problems. Academia is incentivized by publications, and academic scientists to progress through their career and to gain more recognition and higher salary. They don't have to help a single human being live a better life. That's not in their incentives. Their incentive is to publish papers. The more papers, the better. And those papers, most of the time, the vast majority of time, do not translate into anything that's useful to humanity. So we need to change the incentivization model.
Yeah.
Pharma is not interested in preventing or curing diseases. They're interested in managing disease. Their business model is to manage the disease so that they can monetize on that management. If pharma was able to cure someone with arthritis, well, then they're not going to monetize on that person for the rest of their life. That's not their business model. So let's be really frank here and realize that there isn't really an entity or a system who is absolutely focused on identifying root causes of chronic diseases and cancers and then finding ways to prevent them and cure them. That doesn't exist today, except
Right.
Viome.
That's
So
right.
we're very, very excited to be here. Okay, so this is basically a 5-line summary of what the rest of the talk is about. So I'll start with talking about systems biology and how we need to view the human body very differently in health and disease. We're going to talk about epigenetics and that it's more important than genetics. We already discussed that, but I'm going to focus a little bit on RNA versus DNA because we're an RNA company and everyone else is a DNA company. And that's a very big distinguishing point. We're going to talk about our clinically validated tests, clinical validation, and clear performance of any test should be a minimum requirement. If they are R&D tests, then it basically means that they can produce any results and there is no quality control. We're going to talk about our very large clinical research programs only in humans. We have enrolled more than 16,000 clinical research participants in our clinical research programs. Very proud of that. And those research programs have enabled us to basically learn a lot of the foundational biological phenomena in our body to be able to actually understand what's going on. And then we're going to talk a little bit about use of AI.
Momo, I am going to jump in here. Why, a question that popped up in my head, why DNA versus RNA? And also epigenetics, is that also known as gene expression? I think you're going to go into that.
I am going to go into that soon.
Awesome.
Okay. So let's first discuss this topic, which is the human holobiont. So you may hear this technical term holobiont, or you can maybe hear that we are an ecosystem. We have evolved. This is not by chance. This is not because microorganisms are parasites on us. I mean, there are some parasites, but for the most part, microorganisms and us have co-evolved. They are beneficial to us, we're beneficial to them, and I'll show you some extreme benefits that microbes provide to us. But this is the summary, basically, that we encode our human genome that we inherited from our parents encodes for 23,000 genes, which means 23,000 functions. The microbial genes that are in one person number more than 10 million. Of those, several 100,000 are actually expressed at any given time. And so if you just roughly say that 230,000 microbial genes are expressed at any given time or so, then there's 10 times more microbial genes that are currently expressed and functional on our body than there are human genes. So we are an ecosystem where we play a minor role. Our genes play a minor role in our physiology and our health and disease. And so that's why we need to introduce this concept of systems biology. And systems biology goes beyond microbiome. Systems biology means include all systems, all parts of the body that contribute to health and disease. And so systems biology looks at not just our genetics, but looks at our immune system, our hormones, and gene expression. But it also looks at microbes in terms of who is there and what they're doing. It looks at exposures to any kind of a toxin, including mycotoxins. It includes any kind of environmental exposure to heavy metals. It includes, of course, nutrition and what molecular ingredients we are bringing in via nutrition and which ones we're missing. Because you may be eating a peach today thinking, ooh, this peach has a lot of good nutrients for me. But if it was grown on a farm that's completely exhausted, it may just be a sack of water with some flavor in it that's not really as nutritious as it used to be. So systems biology is really this holistic view of the human body that's influenced by many, many different internal and external factors. And we really need to look at it that way in order to understand it.
Excellent.
Okay, here's the answer to your question, RNA versus DNA. This is literally the most commonly asked question. Why are you guys studying RNA and analyzing RNA when everyone else is doing DNA? That's so unusual. So I want to first mention that RNA is a molecule that's very similar to DNA, but it's actually a copy of DNA. So it has the same sequence as our genes, but it's actionable, meaning DNA is a static molecule that doesn't change much, and it is basically inactive. It doesn't do anything. You can have a gene for something, but if it's not expressed into RNA, it doesn't serve any purpose. It's just it's a physical object that's floating in the cells. When DNA is copied into RNA, that means that gene is activated, and the RNA is then converted into a protein. It's called translation. RNA is translated into a protein, and that protein is the one that carries the function of a gene. And so until RNA is copied from DNA, DNA is useless, basically. Now, let's talk about a little more about RNA. So this is something that should resonate with most people, and that is, if you look at 3 organs in a human body and you sequence their DNA, you're going to find that their DNA is exactly the same. Every single cell in our body carries the same DNA that we inherited from mom and dad. Every single cell. So how can brain be so vastly different from liver and kidney in terms of what it looks like and the function it performs? It is as far away from a kidney as you can imagine. The only difference is which genes are actually expressed. And so really, this is one of the most important takeaways from this entire presentation, is that DNA simply means a potential. When you're born, you have a potential. Every cell in your body has a potential to be a brain cell or a liver cell or a kidney cell. It also has a potential to stay healthy for a very long time, or it has a potential to be sick. But what actually happens, you won't know until you measure the gene expression. And this gene expression is what we measure. We actually quantitatively measure gene expression of both human and microbial genes using our analytical methods. So we don't build models to assume anything. We don't make any assumptions. It's literally a quantitative measurement using a clinically validated test. So all these organs have the same DNA, but very, very different RNA. So for those that are curious, you can read more about that. Here's the second slide that's also very important. DNA does not make a difference between healthy and sick state. So here on the left side, I'm showing very graphically the intestine of a person with Crohn's disease, where on the left side, one day, they have a normal intestinal lining. They have been diagnosed with IBD, but at this moment, they're in a remission and they're perfectly healthy. They can go to work, they can eat whatever they want, they don't have any pain, they have a normal life. But a week later, their intestine inflames and they go into something called a flare. And that intestine becomes super, super inflamed. They are in pain 24 hours a day. They cannot eat anything. They have 15, 20 diarrheas per day. Their life just turned into misery. The difference between these two in DNA is none. There is absolutely no difference between DNA of the human cells on the left side and the right side. It's the same DNA that you were born with.
Right.
The difference in the microbial community composition is exactly the same. There is no difference, meaning the microbial community, when you're healthy and when you're sick, as you go through those cycles, it stays the same. It's not like you picked up a pathogen and it causes your flare. It's the same microbes. The difference is what genes are microbes expressing to initiate this process of flare, and how the human cells are reacting to that new environment with their own changes in gene expression. So the genes that are expressed in these two stages are very different, and that has been measured now for many years. I'm just listing this one publication from 2016, but there are others showing very different gene expression profiles. So you can tell the difference between a healthy state and a sick state very easily with RNA. It's literally black and white, or in this case, blue and red. With DNA, you can sequence it as many times as you want to. You will never see a difference because there isn't one. And so these are really key takeaways that we wanted to measure RNA from way before Viome was formed, because it is relevant to our health and disease.
Yeah, that's an amazing takeaway. I didn't mean to interrupt you. Go ahead.
No, if you have a question, let me know.
Yeah, actually, I do. So gene expression, obviously, you just went through that, is clearly very important to understanding both health and disease. So how are we measuring gene expression? Because that's the main thing that we do, yeah?
Yep, great transition into the next slide. So we have now published 3 papers in peer-reviewed literature, and those are listed on our website, that describe our clinically validated tests for saliva, stool, and blood. So you can get a lot more details from there, but this is a summary, and I'm just going to walk through it very quickly because this is kind of really highly technical and not really super important for most people. But basically, we take any clinical sample, stool, blood, or saliva. We perform sample lysis, which means we destroy all cells in that sample. We blow them up physically, microbial cells, so fungal cells, bacterial, archaeal, viruses, and human cells. We just physically destroy them. And then there's DNA and RNA that's floating in space now in the solution. We purify those two, and then we remove the DNA. So we physically remove DNA from the sample. We actually degrade it enzymatically using an enzyme called DNase. Now we have total RNA from all cells that were in that sample. And now we are going to remove these red RNAs. The majority of RNAs in any sample are what we call non-informative. There are ribosomal RNAs and some other RNAs. There are very few of them, but they're highly abundant. So we just remove those physically. And then we sequence the remaining RNAs using a quantitative directional library sequencing method on Illumina sequencer. So this is state of the art. We have patents awarded for this. So this is very unique technology. One of the biggest advantages that maybe people will take away from this slide is the line on the bottom that our method is more than 1,000-fold more accurate in terms of creating noise in the sample than other methods. So we have at least 1,000-fold lower noise level, and this is both patented and published. And so this is a very important thing to remember, that most of these next generation sequencing technologies generate about 1% noise, and most of the molecules that you measure are below 1%. So you're trying to measure a signal that's lower than noise. We have moved that noise down to 1 part per million. So most of the signals that we're measuring are truly above the noise. And that's really a foundational technological advantage that we have, is not only are we measuring RNA, which is relevant to human health and disease, but we're measuring in a far more accurate way. And again, this is published and the patent has been granted and available, so you can read, you can geek out all you want on this technology, but we got to move on.
Yeah, absolutely. I do have one question though. So so that slide is amazing. Like that you're actually able to put, all of our sequencing just in that one line. But when you say other commercial genomic technologies, does that include other microbiome tests?
Yeah, that includes, I am not aware of any microbiome test out there that is using technology, the technology that minimizes the effect called barcode hopping or index hopping that we have invented. And so, yes, I believe, I don't study other companies in detail, but I believe that none of them are using this kind of technology, which means they have to deal with a lot of noise. And that really disables you from finding the signal and finding the really relevant features. Now, the bigger fundamental problem with other technologies is that, remember, they're studying DNA, which means all they can do is tell you who is there. They cannot tell you what they're doing. And we're going to actually get into that right now. This is actually the most important and the most exciting part of the presentation for me, because now we're going to get into functions of the microbes and not just the composition of the microbes. And I'll go over that right now. So to me, the biggest advantage of Viome is that we're actually quantifying molecules that are relevant for human health. If you don't quantify RNA, if you just sequence DNA, and DNA can make you healthy, the same DNA can make you healthy and very sick. There is no point. There's very little point. Okay, this is an exciting slide, and the next few slides are super exciting. So hopefully people can take a deep breath and follow me here.
We're going to do it.
So many of Viome's scores include the word pathway. And I want to talk about what pathways are, and want to define them, and want to show you how important, crucial these pathways are. So a simple pathway is basically a system of chemical reactions or biochemical reactions that a microorganism or a cell would perform in order to do something. And so here, we're going to make beer. And so if you've made beer, or if you know how beer is made, you need to have two components. You need to have a polysaccharide that is digestible by brewer's yeast. So people use typically barley. And then you have to have brewer's yeast, a microbe. And if you provide that polysaccharide from barley to brewer's yeast, and in water, of course, brewer's yeast is going to turn on the genes, about 15 different genes, that allow it to break down the polysaccharide into monosaccharides that enable it to break down that monosaccharide into ethanol at the end and harvest chemical energy for its life. So the way microorganisms live is that they harvest chemical energy from some kind of a substrate, in this case polysaccharide, to convert it to a product, in this case ethanol. And so a pathway is basically a system or a cascade of biochemical reactions that go from a substrate to a product. And in this case, brewer's yeast is going to make us beer from a polysaccharide found in barley. And so that's a key takeaway, and I'll go a little deeper into the next slide. But the takeaway here is that if you do not provide the polysaccharide to brewer's yeast, so let's say in a vessel you want to make beer and you give it barley and you give it brewer's yeast, it will make you beer. But instead of barley, if you provide chicken, if you blend chicken and put chicken in there, will brewer's yeast make beer? Absolutely not. It will not be able to do that because it does not have the raw material, the polysaccharide, in order to make that ethanol. The chemical reactions that take place convert a polysaccharide into monosaccharide into ethanol. They can start with chicken. And so this is a key sort of an aspect of Viome in that we are modulating the biochemistry of microorganisms by either providing them or with substrates or withdrawing those substrates found in our foods. That's basically what it is. If our clinical research studies say brewer's yeast makes ethanol under certain conditions and ethanol is bad for you, we're going to basically tell the person stop eating barley, because since you have this specific strain of yeast that's producing these genes, activating these genes for converting barley into ethanol, you need to stop eating barley, and therefore you won't have ethanol, which is bad for you. Does that make sense?
Yes.
All right, now we're going to go a little deeper, but this is really, really fascinating because it ties everything that we're doing with something that people have heard in the news. So people have heard in the news that some people get COVID once and then they're protected, or they get vaccinated once and they're protected. But other people repeatedly get COVID, and some people get long COVID. Why is that? So let's dive into this. Is the most detailed microbial physiology and human physiology. So stay with me, please. This is amazing. So if you have in your colon microbes that can make something called butyrate, and probably many people are familiar with butyrate because it's sort of a keystone molecule made by microbes. It's a byproduct of microbes, just like ethanol is a byproduct of yeast fermenting barley. Butyrate is a byproduct of bacteria consuming fibers. So when we take a prebiotic,
Mm-hmm.
bacteria in the colon will convert that prebiotic, which is essentially a polysaccharide. They'll convert into monosaccharide, which is glucose, and they will go through these steps. This is a pathway that leads from glucose to butyrate. So that's a pathway, a butyrate production pathway. And all of these steps here are executed by products of genes that are activated, and we can measure their activation level by quantifying RNA molecules. So we can quantify the level of butyrate production. Now, this is happening in the colon. That butyrate will then translocate into the bloodstream. And now our T cells, which are the most important part of our immune system for killing viruses, such as SARS-CoV-2, the butyrate is a chemical messenger that tells a T cell that has just encountered a SARS-CoV-2 virus to actually convert into a memory T cell. And it's doing that via its own pathways. You can see here I'm drawing a regulatory pathway where butyrate activates certain proteins on the surface of the T cell, and via these so-called signal transduction pathways, the human T cell regulates its own gene expression, meaning it turns its own genes that convert it into a memory T cell. And now, next time you encounter SARS-CoV-2, that memory T cell will be there and will protect you. But that memory T cell will not exist if your microbiome did not generate this butyrate. So here's how we connect food with our immune system. If you have the right microbes in your colon, if you give them specific fibers from your diet, they will produce butyrate. Butyrate, during an active infection, will stimulate so-called CD8 T cells to then convert into memory T cells. And these memory T cells will not die. They will stay in your blood for decades, essentially. And they will prepare you for the next infection by SARS-CoV-2 and prevent COVID. And this is such an amazing thing that this happens. And we didn't know this for decades. This mechanism was just discovered recently and published in this publication that I listed here. And so this is just one mechanism where nutrition and specific molecules in your food actually enable you to live a better life by being now resistant to COVID, whether you received it as a virus or as a vaccine. And so my takeaway for my family and friends is, if you get sick, make sure you eat fiber, follow your Viome recommendations, and definitely consume all fibers. The really sad thing is that in developed countries, so-called, right, we use processed foods that have actually removed fiber, which means that we were deprived of these fibers, which means that we got COVID repeatedly. So the COVID burden in developed countries is much higher than in developing countries, because in developing countries, they actually consume fiber. And there's evidence that people who were exposed to coronavirus that was similar but different from the SARS-CoV-2 10 years ago, they actually had enough residual resistance in third world countries to where they were more resistant to COVID.
Wow.
And so these are all amazing things that are happening in the human body, and Viome Science is able to make all these connections and connect them with foods and recommend foods that are actually modulating your microbiology in your colon and then helping you live healthier lives. How cool is this?
That is so cool. And also a good takeaway, make sure you're getting enough fiber in your diet as well.
Well, this is one of the many reasons you want to get fiber in your diet. And I just want to mention here that, we have local health stores here in Seattle, and one particularly that we like is called PCC. Mm-hmm. However, their bread section has about 20 or 30 different kinds of artisan breads. And while most of them have a label like whole food, wholesomeness, or whole grain, or things like that, in words, it seems like they would be whole grain. Not a single one has whole wheat as the first ingredient. Not a single one. Every single one of them has highly processed white wheat that's rid of the fibers as the main ingredient. They literally sprinkle some whole grains on top surface, and they call it whole grain bread. Yeah. That's a complete marketing scam. Yikes. And people really need to educate themselves and say they need to go and read every ingredient. And so just go find bread that first ingredient is whole wheat. And if you can't, go buy whole wheat flour and just make the bread at home. It's very easy. All right. Any questions?
Nope. I'm ready for the next slide.
All right. So here, all I'm showing here is that if you do not provide your microbes with this micronutrient called fiber or prebiotic, they will not make you butyrate. The butyrate will not signal our T cells to convert to memory T cells. And therefore, when you encounter COVID, you will get sick again, and so on. All right. So hopefully that was awesome, because I love it. Okay, so here, now we go into a little bit of how do we translate all of this math and biochemistry that we measure into something that a consumer can actually understand. And so on the left side of this graph, so if you're already a Viome user, you will know that we provide somewhere in the order of 60 or 70 scores, depending on which kit you buy. And these scores are shown like this on the right side: heart and metabolic health, immune system health. But if we open the hood and look what's under, how's that score computed? Well, on the left side, we broke that down into a publication. So if you follow this publication, it's a peer-reviewed paper where we show exactly how we create these scores and how we use these numbers on the left side to compute the score for each person. So if you look at the table on the left, it has two columns. The loadings or weights are the mathematical formulas that are applied to the KOs on the right side in the right column. So the KOs are basically microbial functions that perform certain things like urease production or butyrate production or uric acid degradation or whatever it is. And so we basically measure these KOs using our lab technology. We apply the math on the left side and we compute the score. So this is all 100% objective, mathematically achieved. There is no person sitting there saying, gee, your microorganism is high, so you should probably, I don't know what, right? So that's really the beauty of this, is that it's very objective and it's all computed. And as time goes on, these mathematical formulas will get more comprehensive and more accurate. But right now they're doing really well. And we now have published several papers showing how well they're doing. I'll just throw in one more geeky slide here. Here's the gut score for uric acid production pathways. So we're going back to the pathways score, right? Oh yeah, let me show you on the previous slide. This scroller thingy is in my way, but you can see on the bottom left the size of the study, how many people it involved. Here on this slide, you can see here that these are data on the left side. The loadings on the left side were computed from 66,000 people. I mean, if you go look at the top literature produced by top universities, you will see very small studies that are typically in under 100 people. And look at our study, 66,000 people. And so from 66,000 people, we can compute very, very accurately the contribution of every single microbial function to anything that we're looking for, and map them on the physiological map of the bacteria on the right side, the purine degradation. And all of this has been published as well. So go geek out if you'd like. Okay, so how does all of this math and all this biochemistry we're measuring, how does it actually correlate with phenotypes or diseases? So let's talk about that a little bit. This is our most recent paper that is under review right now. It should be published in the next couple of months. But let me show you, this is actually published as a preprint so you can see it, but here's a little preview. So on the left side, we have 8 different scores. These are all pathway scores measured in the gut microbiome using our technology. What you see in the 4 panels is how does that score correlate with IBS constipation and IBS diarrhea. So let's just focus on the middle 2 panels, IBS-C and IBS-D. So for those who are familiar with IBS, people will generally consider them to be IBS. Well, it's IBS, right? But IBS turns out to be 2 very different diseases. Well, there's more subtypes. There's actually a mixed subtype and there's an undetermined subtype. But let's focus on the 2 major subtypes, which are the constipation and diarrhea. And look at the scores. If we look at the GABA production pathway, so GABA is called gamma aminobutyric acid. It's a neurotransmitter that's produced by microbes. And you can see here the line, the vertical line is actually means that if the data fall there is no effect of the score there is no association of the score with that phenotype. But if it's either to the left or the right, there is an association. And so you can see here that poor GABA production is associated with both IBS-C and IBS-D on both sides. So GABA is very important, or it's very strongly associated with both IBS-C and IBS-D. We then slide down to lactic acid production. And we can see here that lactic acid production score has opposite effects for IBS-C versus IBS-D, and both are mathematically or so-called statistically significant. So this is not by random chance. This is on a very large cohort, and this is statistically significant. And their disease level and type was actually measured using Rome 4 clinical standard, which is the standard that everyone uses. And then if you slide down to methanogenesis pathways, you can see that methanogenesis pathways also have the opposite effect in terms of IBS-C and IBS-D, but they also have an opposite effect of the lactic acid production pathways. So we can see here that GABA has same correlation with IBS-C and D. Lactic acid has different, and then methanogenesis has different and opposite to lactic acid. These are all molecular features that the only way you can get is by measuring RNA. There is no possible way that you can measure this with DNA. It's not possible because the DNA will be the same in all of these. And so this is really showing the power of our technology, but also how deeply we can profile someone at the molecular level. And now a person who comes to Viome who has IBS-C will get different recommendations from a person who comes to Viome who has IBS-D. And the reason is because their molecular scores are different. We are measuring different biochemistry. And that's really quite an incredible thing that we can dig so deeply into the microbial physiology and then convert that or translate that into specific nutritional recommendations. Very, very, very proud of this work. It's been 14 years, and this is amazing.
That is awesome. Momo, I think I have this paper. Is this the suboptimal activity paper on IBS or not? That's not that one?
Yeah, that's one of them. That's one of the papers, yes. So go ahead and post it.
Yep. So if you want to close that window down after you click on the link, you can click the close button on the bottom. And there we go. Yes. So please continue on.
All right. So now the question is, how do we actually convert all this molecular knowledge into specific recommendations? And so we have a clinical nutrition team, which you guys know how awesome they are because they've been in so many webinars. And so we created this platform called Nutrition 2.0. And Nutrition 2.0 is nothing more than biochemistry and math. And we talked about biochemistry, we talked about math. Maybe I'll talk a little bit more about it, but biochemistry means we are quantifying the expression level of active genes in all microbial cells and human cells. With math, we're creating mathematical formulas that convert those measurements that we make on the biochemistry side into scores. Mm-hmm. And now we're using those scores as well as the subtype, the type of phenotype that a person has into nutritional recommendations. So the takeaways for Nutrition 2.0 are that it's 100% data driven, meaning there is no person who is inputting anything. It is solely based on the data, and it is 100% objective, meaning mathematics, 2 plus 2 is always 2 plus 2. There is no, oh, for this customer, we're going to say 2 plus 2 is 3. No, it's always exactly the same. Every customer gets completely computed nutritional recommendations. And therefore, it's highly personalized. We have 500,000 customers. We have 500,000 recommendations because the computers do not know or care about ketogenic diet or Mediterranean diet or anything like that. They're specifically feeding or withdrawing specific micronutrients for each person in order to modulate their biochemistry. Okay, its goal is to improve health span, lifespan, and slow down aging. And then it is self-learning and continuously improving. So that math that I showed you, we either continuously or in batches continuously improve that math over the years, so that next year our recommendations are going to be better. Next year they're going to be better as we do more clinical research. Of course, clinical research costs an insane amount of money. Mm-hmm. You know, one project that we carried out in the US and Japan for learning what influences the blood sugar levels from our microbiome cost us several $1 million. And so these clinical research studies are extraordinarily expensive, and we have to survive as a company, so we have to balance it all that. But one day we're going to be able to carry out all clinical research studies needed to understand fully the human body and all phenotypes and all diseases, and then really reveal everything. It's just a matter of time. Okay, this slide is sort of just an overview of what it means to be a Viome user. And so just in summary, most people probably know this, but we've applied Nutrition 2.0 to more than 500,000 customers in more than 100 countries. A person will collect samples. The best kit, obviously, is the one that collects stool, blood, and saliva, which is this one right here. We process those samples in the lab. We discuss that. From these 3 samples, we generate just over 20,000 data points, meaning we measure 20,000 things versus when you go for a blood draw and they measure 10 things. We're doing 20,000. And then we apply mathematical algorithms like we discussed, and the person receives their personalized recommendations, which are here. And then every 6 months, they repeat the process and keep fine-tuning their body. So that's the process. Okay, the last part of the presentation is really how well does this work and how are we measuring how well it works. So I'm going to go through this fairly quickly, but happy to answer any questions. Here are scores, gut health scores and oral health scores on the right side. And we've measured now how well our customers do from the moment that we measure their score the first time and then the second time after a few months. And you can see here that 15 out of 17 gut health scores significantly improved. This is statistically significant, so it's not by random chance, after people followed the Viome diet for at least 3 months. And oral scores, 10 out of 12, improved with statistical significance. So that's really, really important that we can, in fact, with foods, modulate the functions of the microbiome because those functions are quantified using these scores. Yeah. And so that's really the fundamental thing. Can you actually do something about it? Because today, if you get a diagnosis of something, but they're like, well, you have IBS, sorry, but we can't do anything about it. Well, that's not very helpful. So we have to translate that knowledge into recommendations. Mm-hmm. I'm going to skim this because this was published in a peer-reviewed article that Lisa can share. It's basically, we ran 4 interventional trials for IBS, depression, anxiety, and type 2 diabetes. And we call these gastrointestinal mood and metabolic health studies. And these were actually following Viome customers through their journey and recording their either symptoms based on clinically validated surveys or their molecular signs, as in the case of type 2 diabetes. And we show very significant improvement in all of these scores and all of these signs. And so please read that paper, educate yourself. These are not randomized controlled trials, so they're not as robust as a randomized controlled trial would be. But given the level of improvements, we felt compelled to publish this because this is really important. And these are very prevalent diseases that are growing in prevalence. And this just is good to know. All right, so here's another depression study that we are currently going through the publication process, just showing that we were able to improve the mood of people using a clinically validated survey called PHQ-9. We got statistically significant improvement in people who both took our supplements and followed our nutrition relative to customers who did not take our supplements and did not follow nutrition. So that's very important, both nutrition and supplements to follow. Then we have some preliminary results. Note that these are all preliminary results from our randomized control trials. So we're running 4 randomized control trials, and these are just first look. And so you can see here, this is a randomized control trial for mood, where the clinically validated survey called PHQ-9 is used. It's a very clinically acceptable survey. And we can see here in blue lines, in blue bars, are the improvement of placebo. These participants received placebo pills, and they did improve. And in fact, it is known that mood can be improved with the placebo effect. But if you look at the yellow part, which is Viome nutrition, you can see that the improvement was much higher. And in fact, we have a very strong trend towards significance, and we have a much stronger improvement with Viome nutrition than with placebo. So we're very happy about that, but of course, we have to keep going. Similar results for gastrointestinal issues. So this is measured with a gold standard IBS SSS clinically validated survey, also blinded randomized placebo-controlled trial. You can see that the placebos improved, but the Viome nutrition participants improved even more by 76 points more than the placebo, which is considered clinically significant. Anything over 50-point improvement over the placebo is considered clinically significant. And the p-value is very low. So this is we have very high confidence in this result, but we're continuing with the trial to get larger numbers. And last but not least is our impaired glucose management. So remember, well, you don't remember, this is not a part of this presentation, but we invested heavily a few years ago into understanding how different micronutrients contribute to blood glucose spikes and how they're modulated with gut microbiome, and how can we modulate gut microbiome and nutrition. And this is now basically the fruit of that labor, where on the left side, participants were measured with HbA1c tests at Quest Laboratories, and we can see here that we are observing very good preliminary results where the yellow, which is the Viome nutrition participants, improved their HbA1c, reduced it by a lot more than the placebo participants, and the p-value is very low, so we're very confident in this result. And on the right side, we're looking at the fasting glucose, which is another marker of prediabetes and type 2 diabetes, and we can see that actually the placebo fasting glucose went up in 3 months, whereas in our intervention, the glucose went down. This is trending towards significance, but we're continuing with these studies. And that's it.
That's amazing. Like everything that you're showing is that food can be considered medicine, right?
Absolutely. I have to keep reminding people that nature has already figured all this out. We have tens of thousands of people currently living, they're living proof, who are in their 90s, who literally have a normal life like a 20-year-old. They can cook for themselves, they can clean, they can dress, they can walk, they can perform their daily tasks. You can have a conversation with them. They don't have any disease. They don't have to take any medications. There's tens of thousands of them, and they're concentrated in so-called blue zones, but there are many others that are not in blue zones. Yeah. And these people have basically fine-tuned their nutrition and lifestyle, and they have avoided toxins and plastics and all this, developed country toxic burden. and they've been able to fine tune their lifespan to where they're healthy well into their 90s. And all we're doing here at Viome is trying to understand how nature has figured this out, and then empower individuals to have that kind of life without having sort of moving to a blue zone and living that lifestyle.
Awesome. Thank you so much for that, Momo. I do have a few questions that I would
All
like
right.
us to try to get to. And apologies to everybody hanging out. I am single-handing this today, so there's just me, so I couldn't jump in there and start answering questions. But we are going to like pull these questions that Momo can definitely answer for you. So let's do that one. So I'm going to pop this up for you, Momo. You can see that, right? On the screen for
Yes,
Melanie.
yes, I can.
So she had a question about the information within the Viome test. It's a slice of time in our lives and actually carrying enough pertinent information to understand our current state. So she just wanted to understand what is it, what data that we are giving to them. How does a single Viome test give that to us?
Well, a single test is very valuable in that it makes recommendations for how can you tune your physiology today, given the state of affairs today. If after 6 months it's different due to various reasons, it'll give you a different recommendation. And that's perfectly normal. That's been observed in many studies that the gut microbiome and our gene expression changes over time. So there's value in both, but really the most valuable thing to me is today, what do I need to do tomorrow? What do I need to cook tonight in order to improve my physiology? That's the most important thing. Now, if someone sees that their microbiome changed very dramatically in the next 6 months and not necessarily for the better, that may mean that they have some influences in their life that they're not aware of. And that can be either most typical would be antibiotic use that you're not aware of, such as in non-organic meats. It can also be, just so that we're clear here, if you go buy meat at the grocery store and it says all natural means it's a legal term that means that you cannot treat that meat with any chemical after the animal dies. Until the moment that animal dies, you can pump them with antibiotics all you want. You can still label it as all natural. That is really shameful, by the way, but that's what it is. So you can be consuming antibiotics without your knowledge. You can be consuming all kinds of preservatives that act as antibiotics, because that's what they are, in your foods if you don't read your labels, right? So read your food labels. Obviously, the best food is whole food, plant-based, organic diet that is locally sourced, but not everyone can do that, and that's a pretty major shift. So just do the best to read the ingredients. One example that I often give is people will be recommended, let's say, sauerkraut, because it's a probiotic. It's a probiotic, right? A probiotic-rich diet. You go to a grocery store, and whether you go to a shelf or to a refrigerated section, you are in danger of buying sauerkraut that was fermented with bacteria, but then they added preservatives, typically sodium benzoate, to kill all those bacteria to stop further fermentation. So now you're eating sauerkraut believing that you're eating probiotics, when in fact those probiotics have been killed, first of all. And second, you're eating that massive amount of sodium benzoate that they added to kill those probiotics. You're consuming it, and now you're killing your own oral microbiome and gut microbiome. So instead of getting the positive effect of these probiotics, you're actually hurting yourself. This is one of many ways that food industry has figured out to keep products on the shelf for longer, to keep lowering their manufacturing costs and all that. So be careful what you read and how much you spend time on. You can also have a gastrointestinal issue where you had diarrhea for 5 days, and you can change your microbiome quite significantly. You could also be exposed to all kinds of environmental intoxicant. You can be exposed to mold in your workplace or your home, and that can really affect your microbiome. So there are other things. So this, over time, retesting is very important so that you always get up-to-date information about your body, and maybe hint at, there's more to this than just what I think is going on. I need to dig deeper into what am I being exposed to?
Yeah, absolutely. And you bring up a really good point about retesting, in that this one slice, as Melanie said, this one slice in time, yes, is important for now, but your body is dynamic. It changes, especially if you're going to follow these recommendations regularly. You can check back in and see how you're doing and see what needs to be tweaked. These are not recommendations for life. You don't have to stay away from this food or eat only this food for the rest of your life. Your body is dynamic. Yeah,
Yep.
absolutely.
Yep.
I do have one question here that I do want to pop up because it just tells the difference between what we do and what we don't do. Carmen was asking about food intolerances and food allergies, and she's confused. She says that the list of foods is bad for me were almost completely different than those tests, and if I followed all of them, I'd have nothing left to eat. So she wants to know.
Yeah. No, this is a great question. This is a great question. Food intolerance. So IgEs, I have not seen very good utility for it unless you're truly allergic to something and you want to confirm with that test. But IgE response would be sort of violent and pretty instant after a meal. So I'm not super. I think it's a good idea to test if you can, but if you cannot afford to test it, I would put that aside. But IgG intolerances, I think, are super useful, and they're very complementary to our tests. Now, what if you observe two different results? So let's talk about that.
Yeah.
So this is all my opinion. There is some literature backing this, but this is still a very new area of research. But to me, food intolerances should be followed very strictly, meaning if you have moderate or high food intolerance, then I would avoid those foods. Now, let's also, first, before we talk about food tolerance results, I see a lot of scamming companies out there that are performing food intolerance testing, especially those that say we can give you 800 food intolerances from your hair. I am not aware of any analytical technology that can quantify your IgGs in your bloodstream using a hair follicle. I'm not aware of it. I could be proven wrong, but I believe that those are scammers. And there are many other companies who don't have very good tests. The only company that I trust with IgGs would be Everlywell. That's it. That's the one I've been ordering for my family for years. We've seen great results with them. That's the only one I would recommend. Now, let's make sure that we understand what we talked about earlier, which is systems biology. Remember, systems biology is nutrition, microbiome, immune system, hormones, genetics, all of that stuff. Immune system response is a very different thing from what we measure at Viome, and it's very complementary. It's not overlapping. And so, for example, if we say that you should be eating some food because it contains micronutrients that are going to feed your microbiome, specifically your microbiome, in order to produce something beneficial, that is objectively measured in your gut microbiome. We didn't make that up. That is what the chemistry of your body is telling us, right? And we measure that quantitatively. So there is no disputing that in my mind. However, if that food contains some kind of a protein that acts as an antigen for your immune system, and your immune system responds to it, then I would say that should override the benefit that you derive. Now, ideally, and at some point in time, we will offer our own food sensitivity tests, and we will integrate it with our normal tests so our computers can actually provide you with an alternative. So for example, today, if our computer says, well, you need to be taking broccoli because of sulforaphane, but if broccoli is causing you food sensitivity, then our computer would be built in with logic to say, oh, but you cannot take broccoli, so go ahead and stop eating broccoli, but we're going to put sulforaphane in your supplements, right? One day that's going to come to Viome. We're just not there yet. And so you're going to have to manage the results of food sensitivity tests and our results. But what I practice with my family for the last many years, and what I recommend to everyone I care about, is if you have moderate or high sensitivity to a food, get rid of it completely for 3 to 6 months and see if it improves anything. If it doesn't, it was a false positive, and that's common in food sensitivity tests, unfortunately, but it is what it is. But if it improved your condition, great, keep it that way.
Right, exactly. And just so that everybody knows, if you have not taken a Viome test, you can indicate to us in the questionnaire that you have to take with your test foods that you are allergic to and foods that you are sensitive to. So those
That's a great point. Great point. So if you do the food sensitivity test before hours, and then you plug that in, then it'll change our recommendations. Yes. Thank you, Lisa.
That's right. Yeah. So it will obviously, foods that you are sensitive to, it will put onto the minimized list, and foods that you are allergic to, it will automatically put that onto your avoid list. But that's because you have named them out as those are foods that you are sensitive and allergic to. So, but that's a good tool so the other foods can float to the top and you can see the actual superfoods that you can have. So there you go. So that's really, really good. I think we have one more. I want to do one more question here, and we are almost at time. So I do want to have this one because it's about pathways. So Momo, Joanna says the pathway results are very helpful, but I wish the results were explained. Why doesn't Viome include regarding how the system arrived at its conclusions? So we did actually give a lot of explanation on that today, but maybe we could just touch on this before we end off the call.
Yeah. Let me address this. The last part of this. Let me address this. So, we're going to publish blog articles as soon as we can, and we're going to publish, peer-reviewed publications as soon as we can. But let me give you an example of a recent finding. Porphyromonas gingivalis is a very serious pathobiont in the oral cavity, and that's because it can produce toxins called gingipains that have actually mechanistically been associated with Alzheimer's disease. And so one would think that increased Porphyromonas gingivalis abundance would actually be bad, but that is not correlated at all. The abundance of Porphyromonas gingivalis has essentially nothing to do with how many gingipains it's producing. And we've observed this with everything else. And so what this means is that knowing the relative abundance, just the presence of a microorganism, doesn't tell you anything. Remember, measuring DNA is just a potential. The microorganism is there. It encodes the genes for gingipains. But if it's not expressing those genes, then it's of no concern. And that's why we don't display quantitatively what are the relative abundances of microbes, because what we don't want is someone going to take some extreme measures with all kinds of herbs and antibiotics and other things to kill off their microbial community because they think something is high, when in fact they're doing themselves a disservice. If something is high, the only thing that matters is the functions and specific functions, and then we are going to provide you with the nutritional recommendations to try to reverse those. Right. But we don't think it's a good idea to apply all of these sort of nuclear bomb style generic reduction like mouthwashes and antibiotics and all these herbs that will make it worse, basically. Yeah. So I hope that makes sense. And in terms of scores interpretations, please look up our peer-reviewed published article that's listed on our website from last fall about oral scores. We also listed the preprint of our article on gut scores. We literally walk you through every possible step of how we arrive at the scores and how we arrive at the mathematical equation for every score. So I'm not really sure what else we can do there. We've opened up the black box and it's all spilled out. Every detail is there.
I gave the biomelifesciences.com. That's where you can find all of our published papers there. And I do want to do one more thing before we go to make sure that everybody who has not tested with Viome and would like to do that can. I am giving you a special discount code. It's called Difference, and you use that at checkout. You can get $100 off of our full body intelligence test if you use that link and you go to our website. The discount code should automatically be applied in the checkout for you. So you can do that if you like. And Momo, thank you so much for coming here and talking to us about all things Viome, the Viome difference, and all this really scientific stuff that I'm sure I saw a lot of people geeking out about. There were a lot of people like, that's so fascinating. I love that. And so thank you once again for spending this hour with us and giving us this awesome information. You're getting lots of emojis and applause, and I appreciate it.
Thank you. We're very excited to be leading this and working 7 days a week with pleasure.
That's right.
Thank you, everyone.
Thank you very much. We will see you here again next month for our next topical content webinar. Thank you very much for spending the last hour with us. Bye-bye and have a wonderful day.
Thank you, Lisa. Thank you, everyone.